Hi, I am Dr.Shilpa Joy, Assistant Professor of Biomedical Engineering Department, Sri Ramakrishna Engineering College, Tamil Nadu, India. Today, I am going to discuss about a wonderful topic, that is stem cell gene therapy. So, through this topic, we can see a basic idea about what is stem cell and how gene therapy is used in recent biomedical applications to cure many diseases. Shall we move on to the topic? Stem cell gene therapy. So, from the word itself, we understand that it is something like gene therapy, that is, we are manipulating the genes. We know that genes are the basic expression units to create a protein. So, based on the health of a gene, that determines actually whether we are healthy or not. So, that is what we are going to discuss, how you can manipulate a gene to use this technology in therapy, and that is mainly focused on the stem cells. So, the learning points of today's topic is like we can define, basically we can define what is a nucleus, what is a chromosome, then gene, and that is gene is present on the DNA. Now, we can describe the gene therapy, and afterwards we will see some advantages and challenges of this stem cell gene therapy. Cell, we all know that that is the basic structural and functional unit of life, and in the cell we can see a very small part that is called as nucleus. So, inside the nucleus, if you see the cell is actually a micrometer size. Inside the cell we can see the nucleus, and inside the nucleus we can see the chromosomes. So, that is what we are discussing today. So, cell is the smallest unit of an organism, and cells are known as the building blocks of life. So, all the human beings or animals or plants, whatever we say, all are made with cells. So, inside the cell I told you there is a nucleus, and inside the nucleus if we go and see, we can see the bucket of genes or DNA. So, the DNA is actually, the stretch of DNA is called as chromosome. So, nucleus actually it controls all the cell activity. How does it control? It is mainly controlling with the help of the chromosomes. So, this chromosomes, they actually contain the DNA. So, the whole DNA is actually constricted to give the chromosomes. So, depending upon the type of organisms, the number of chromosomes also will vary. So, when we go in detail about the chromosomes, I told you already that chromosomes are made up of single DNA molecule, and that is actually in a constricted form, and this is associated with all type of features what are expressed in your body, and this is in a highly condensed form. So, this is actually a detailed structure of a chromosome. So, we can see that the chromosomes are containing the DNA, and that DNA determines the basic structure and function of the cell. Now, inside the chromosome if you see, that is chromosome, I told you that chromosome is having the full stretch of DNA, and inside the DNA if you see, there are specific location on the DNA which is responsible for the expression. That is, these positions on the DNA are responsible for producing an mRNA and thereby a protein. So, those positions are called as gene. So, here it is written that gene is having a sequence of nucleotides arranged to give expression for the human beings. Now, you can locate the gene, that is, this is actually a summary of whatever I have told now. That is, inside the cell we can see the nucleus, and inside the nucleus we can see the chromosomes, and when on the chromosome you can see all the DNA, and in the DNA if you see, we can see certain locus that is responsible for the expression of the cells, that is nothing but the gene. Those locus is called as gene. Now, if you see in a human body, if you see, you can see mainly 23 pairs of chromosomes. So, totally there are 46 chromosomes, and each you can see pairs, that is 23 chromosomes. In the picture you can see that the size and the nature of the chromosomes are varying. So, all the 23 chromosomes are of not same size and shape, they are actually differing, and you can see a longer chromosome to smaller chromosome here. So, depending upon the type of the organism, as I already mentioned, if you are seeing a dog or a monkey, the chromosome number also will change. Even the patterns in which the chromosomes are arranged, that also changes. So, certain cases one chromosome will be missed from a pair, or some extra chromosome will be there. All these can cause certain syndromes in the human beings. Now, basically there are sex chromosomes that is the 23rd pair of chromosome, which is made up of XX type or XY. XX determine the females and XY determines the male. So here we can see that again the size of the chromosome for XX, both the chromosomes are of same size, but when you go for a male chromosome, the X is bigger and the Y is smaller. So in this way also there is a change in the chromosome nature. So now we can go to gene therapy. Before going to gene therapy, just we will have a few words with about stem cells. So stem cells, from the name itself, it is a type of cell only, but these stem cells are actually premature cells. Mainly the stem cells we observe during the embryonic stage of a organism when it develops. That embryonic stage you can see the stem cells. Even adult stem cells are also there in our body. In many organs we can see that. Basically we concentrate on bone marrow cells because bone marrow is actually a rich source of stem cells. So we are going to manipulate the genes present in these stem cells to cure certain diseases. So that is what is highlighted here. So gene therapy, like as the picture shows here, here we can see that a particular DNA is shown here, and a few people are working on the DNA, manipulating the DNA, so that the problems what is there on the DNA can be cured. Problems in the way that certain nucleotides are misaligned or missing, or some additional nucleotides are present. Due to all these cases, certain problems can arise in the body which will be expressed, and that is actually transferred to the next generation also. So these kinds of problems can be cured with the help of gene therapy. So gene therapy is a medical approach that treats or prevents disease by correcting the underlying genetic problem. It involves introducing genetic material into a patient's cell to compensate for or correct a faulty gene, or to introduce a new gene that provides a therapeutic benefit. So here the manipulation doesn't mean that altering the genetic functions. We can introduce a new gene also into a cell for giving a better expression of the gene. So that is what we are going to say now. In gene therapy, if you see that there is a person standing here, and there are 3 types of gene therapy modes. The 3 types are, the first one is in vivo, then the second one is ex vivo, and the third one is in situ. So as the vivo means what? Means living. Okay, that's the meaning. So in vivo means inside the living organism, ex vivo means outside, and in situ means at the site of problem. So in vivo method, what we are doing is that we will be introducing certain foreign genes or DNA or RNA, even some regulator proteins can also be introduced into the patient body, and then the gene of interest will go and integrate with the patient's DNA and will give the new expression pattern. So for this we can use certain plasmids. Plasmids are extra chromosomal matters what you can see in the lower organisms like bacteria. You can introduce your gene of interest in the plasmid, or else you can introduce this DNA strand directly to a particle, maybe a nanoparticle or any carrier. Then or else you can introduce directly the RNA into this particle. Another concept is antisense. Antisense RNA is nothing but the RNA which is complementary to the RNA present in the cell. So whenever this complementary RNA goes and binds with the RNA present in the cell, the RNA dimer will be formed. Usually RNA is a single stranded molecule, we know. So whenever RNA is forming as a dimer or a double stranded molecule, that RNA is actually inactive. It cannot give rise to any protein, or translation is blocked. In this way, that RNA conversion to protein can be prevented. So if any unwanted protein is synthesized in the body in an excess level, that can be stopped with the help of antisense RNA technology. Now the last item is molecular regulators. These molecular regulators can be any growth factors or supporting factors that will help in the cells to give any expression or grow, differentiate, and all these things. So all these factors can be incorporated into the patient body through the cerebrospinal fluid or blood. So what happens is that these molecules which are introduced into the patient will be flowing throughout the body and will find its own site of action, and they do the action and give the result. So this is what is called as in vivo. So the integration with the patient DNA will happen inside the body. Now comes to the next second item, that is ex vivo gene therapy. In this, as the name indicates, the gene therapy or the manipulation of the genes happens outside the body. So, from the patient, blood is taken, and blood or any tissue type that is taken, and cells are isolated. So, from this blood, cells are isolated, and this cells DNA is actually modified. Some genetic markers can be introduced, certain technologies are there to manipulate the DNA. Even we can use the plasmid DNA to infect this cell, and plasmid to infect this cell. This plasmid is actually having the gene of interest, and that will integrate with the DNA inside the cell. After modifying this cell DNA, this can be again put back into the cells. This already we have isolated the cells. From that we have taken the DNA is manipulated, and that is reintroduced into the cell. Now this cell is nothing but a modified cell. So this cell can be introduced back to the patient. So here the manipulation takes place outside the body, that is why it is called as ex vivo. So these we can isolate, instead of any type of cell, we can isolate stem cell. Stem cells are isolated from bone marrow and other tissues also, or from blood also, that can be used for gene manipulation and then reintroduction into the cell and back to the patient. So this is ex vivo. Now the third one, that is in situ. In situ is the action at the site. So in this picture it is shown that on the leg of the patient there is a tumor. So isolate the like we have to see that portion, that is this part of the leg portion is having a tumor. So what we are doing is that take the genetic material. This particular DNA is having a normal condition which will code for certain normal proteins, or it can code for certain proteins that will fight with the tumor on the leg. So this DNA is actually integrated inside a particle. It can be a carrier, micro or nano carrier, and that combination is introduced into the target. So target is nothing but the tumor site. So directly that is targeted to the site, so that the side, actually in this case side effects are very less, because targeting is happening to the particular site of injury or site of cancer or tumor or whatever it is. In both the other cases, in vivo and ex vivo, all the particles will be circulating throughout the blood inside the body, and it can cause maybe some adverse effects also can be resulted. But in situ, such kind of reactions may not be there. So these are actually the three types of gene therapy mechanisms, that is in vivo, ex vivo and in situ. So we can actually manipulate the stem cells by these three methods. Gene therapy for sickle cell disease. So this is one example where the gene therapy is done. So from the patient, actually the blood was collection of blood forming stem cells. So stem cells are actually collected. So blood forming stem cells, they are called as hematopoietic stem cells. They are actually isolated from the bone marrow of the patient, and those stem cells are actually manipulated here. So stem cells corrected by beta globin gene transfer from viral vectors. So virus, these are all actually virus. So this virus is utilized inside the DNA of the virus or the genetic material of the virus. The gene of interest which needs to be introduced in the patient is aligned, and this virus is allowed to infect the stem cells, that is nothing but the hematopoietic stem cells. So after infection, what happens? The gene of interest is now aligned to the gene present on, or the DNA present on the stem cells, and that can be introduced into the patient by transplantation, by just injection or by other means. You can just introduce the manipulated cells, hematopoietic cells into the patient, so that now the patient will be started to express beta globin gene in the blood cells. So this is one example of stem cell gene therapy. Here somatic cell gene therapy is also there. Again, this is a kind of stem cell therapy only. Here we can see a boy. So this boy is having X-linked severe combined immunodeficiency disease. X-linked, as I told you already, we have two sex chromosome, that is XX and XY. So this baby is having X-linked, that is X chromosome is actually affected, and that was leading to immunodeficiency disease, that is deficiency of certain interleukins in the blood cells, white blood cells especially. So these proteins like interleukins are actually responsible for giving immune response in the body. Since those proteins are not expressed in the boy, he is actually an immunodeficient. So now what we are doing is that the bone marrow was isolated. The bone marrow contains stem cells. So those stem cells are actually isolated from the bone marrow, and so now here we can see that these are the bone marrow stem cells. Now to introduce a gene of interest, we have a vector. So vector is nothing but a carrier which can transfer the gene of interest into the stem cells. So here this is nothing but a viral vector. So in the vector DNA you are actually integrating the interleukin gene. So this blue color is actually showing the gene of interest, that is a good gene which can produce interleukins, that is actually linked to the vector DNA. And this whole particle is actually a virus. So virus used for infection can be a retrovirus. So inside the retrovirus the DNA is actually tagged or linked with the human interleukin gene. So now these viral particles are allowed to infect the stem cells, and after infection what happens is that this particular viral DNA will integrate with the DNA of the stem cell. So now the stem cell is actually a modified stem cell where the bone marrow stem cell is having its own DNA which is integrated with the viral vector DNA, and that vector DNA has your gene of interest also. Now this modified stem cell is allowed to grow in a culture media because we have to increase the number of the cells, so that is allowed to grow, and we will be checking whether the expression of that particular gene is happening or not. So after thorough checking and all these things, these cells are actually introduced back into the patient. So when it is introduced back into the patient will be started to express that interleukin protein that will restore immune function in white blood cells. So white blood cells, we know that they are actually the fighters against the disease. Now this particular gene, that is the gene of the stem cell plus the gene of interest, that combination, that particular gene is called as a transgene. So that transgene is actually a combination of the normal gene plus the gene of interest. So now this boy will start expressing that interleukin protein which will make him to live a normal life. Otherwise he is already immunodeficient. If it is immunodeficient, what happens is that the boy will get sick soon. Now we can see what are the challenges in stem cell culture and therapy. So there are many challenges also associated with this therapy because we are actually manipulating the DNA. There are certain ethical issues arise in this case. So that can lead to certain biomaterial toxicity, that is you are using a new DNA of interest, certain growth factors, as I told you, certain growth factors or chemicals you can add to the patient to control the gene expression. So biomaterial toxicity can be there. Then like you expect actually a good expression, but sometimes there can be a restricted expression so that cell division and expansion may not take place. So usually in our body cells are actually dividing and proliferating. So that may not happen sometimes if you introduce a new gene and if you manipulate the gene. Next one is spontaneous differentiation, that is you are introducing a stem cell aiming that stem cell should be converted into a cardiac cell, but sometimes that may not happen. It will be spontaneously differentiating without achieving all the required features. Next one is genomic instability and mutation. Since you are adding a new nucleotide or you are manipulating a nucleotide, that can lead to certain genomic instability as well as it can result in a mutation which will lead to certain tumor formation or something like that. After that scalability and cost, that is scalability and cost is nothing but if you do this kind of experiments, these are actually highly cost factor is involved. So scalability is also a question mark. And then we can see here immunogenicity and immune rejection, that is you are introducing a new growth factor or a new content into the organism. Sometimes that may lead to immune rejection because your immune system will be active to fight against a foreign particle. So that can lead to some immune rejection also. Next, functional maturation and integration, that is you are expecting for a proper maturation of the cell and integration of the DNA into the stem cell, but sometimes that may not happen due to certain factors, that may not happen.
Then last I told you like legal and ethical consideration, that is whenever you are manipulating a DNA, there can be a question like you are manipulating the organism, so many aspects are there. So ethical and legal considerations are also there when you think about the gene therapy in stem cells, because stem cells are actually the basic form of the cell which can differentiate into any type of cell. So if you are manipulating the stem cells, that can lead to formation of a modified organism also. So that is actually a question mark which raises the ethical and legal considerations. Hope the topic is interesting. This is actually a very modern and innovative technology where we can manipulate the DNA itself. So for many diseases, medicines are not available, or that medicines are not giving any proper response. So in such cases, we can go for a gene therapy method which can introduce certain resulting changes in the human body. So the stem cell gene therapy especially is a modern innovation where stem cell itself can be changed. We know that stem cells are the basic form of the cell which can lead to any type of cell type in the body. So if you are manipulating the stem cell itself from the primary stage itself, that problem can be alleviated and we can create a new environment in the cell. So hope this topic is highly relevant for the new biomedical era. Thank you.